Let us go through an overview of Chapter 3 of Biology, that is photosynthesis. Photosynthesis is the only process on Earth by which solar energy is trapped by green plants and converted into food. This process is depicted by the chemical equation as shown. Basically, synthesis of carbohydrates, that is glucose, from inorganic materials like CO2 and H2O with the help of solar energy trapped by pigments like chlorophyll. Chloroplasts. These are mainly located in the mesophyll cells of leaves. Each chloroplast is bounded by double membrane. Inside the membranes is found a ground substance, the stroma. Inside the stroma is found a system of chlorophyll bearing double membrane sacs or lamellae. Two predominant type of chlorophylls. Chlorophyll A and chlorophyll B differ in nature of groups. Chlorophyll A has a methyl group, that is CH3, while chlorophyll B has an aldehyde group, that is CHO. Chemically, chlorophyll molecules consist of two parts head of tetrapyrrole, the porphyrin ring, and a long hydrocarbon tail called pythol attached to the porphyrin group. Nature of light. Light travels as stream of tiny particles called photons. A photon contains a quantum of light. Light has different wavelengths having different colors. Visible light lies between wavelengths of ultraviolet and infrared. Absorption and action spectrum. The amount of light absorbed at each wavelength can be shown in the form of a graph. It shows different curves at different wavelengths. Such a curve which shows the amount of light absorbed at each wavelength is termed as absorption spectrum. Denotes this graph of absorption spectrum. The relative rate of photosynthesis at different wavelengths indicates close relationship with absorption spectrum of chlorophyll A and chlorophyll B. This curve that shows the rate of photosynthesis at different wavelengths is called action spectrum. The figure 12.4 shows the action spectrum of photosynthesis. Mechanism of photosynthesis. Here, bacteria use H2S and CO2 to synthesize carbohydrate as shown in the equation below. Figure 12.5 shows the photoexcitation of chlorophyll A. Thus, Hill reaction proves that in photosynthesis, oxygen is released from water. Electrons for the reduction of CO2 are obtained from water. Light reaction. In light reaction, solar energy is trapped by chlorophyll and stored in the form of chemical energy as ATP and in the form of reducing power denoted as NADPH2. Reaction center. The light absorbing pigments are located in the thylakoid membranes. These are arranged in clusters of chlorophyll and accessory pigments along with special type of chlorophyll molecules P680 and P700. The letter P stands for pigment and the numbers 680 and 700 for the wavelengths of light at which these molecules show maximum absorbance. Photosystems, first and second. The thylakoid membranes of chloroplast have two kinds of photosystems, each with its own set of light harvesting chlorophylls and carotenoids molecules. Chlorophyll and accessory pigments help to capture light over larger area and pass it on to photocenters. Photosystem second. This system brings about photolysis of water and release of oxygen. In this act, when PS2 absorbs light, electrons are released and chlorophyll molecule is oxidized. The electrons emitted by P60, PS2 are ultimately trapped by P700, that is PS1. Figure 12.6 shows the ATP synthesis through chemiosmosis. The oxidized P680 regains its electrons by the photolysis of water as denoted below. Oxygen is given out as a byproduct by photosynthesizing plants. Protons, H positive, accumulate inside the thylakoid resulting in a proton gradient. Photosystem first. When light quanta are absorbed by photosystem first, P700, energy-rich electrons are emitted from the reaction center. These flow down a chain of electron carriers to NADP along with the protons generated by splitting of water. This results in the formation of NADPH. Photophosphorylation. Formation of ATP in the chloroplast in presence of light is called photophosphorylation. It takes place in two forms: cyclic photophosphorylation. Elimination of photosystem first causes electrons to move continuously out of the reaction center of photosystem first and back to it. Second, non-cyclic photophosphorylation. It involves both PS1 and PS2 photosystems. In this case, electron transport chain starts with the release of electrons from PS2. In this chain, high energy electrons are released from PS2, do not return to PS2, but after passing through an electron transport chain reach PS1, which in turn donates it to reduce NADP positive to NADPH. Figure 12.8 depicts the same. Dark reaction. Carbon fixation occurs in the stroma by a series of enzyme catalyzed steps. Molecules of ATP and NADPH produced in the thylakoids, that is light reaction, come into the stroma where carbohydrates are synthesized. With the help of radioactive carbon, it becomes possible to trace the intermediate steps of fixation of 14CO2. The various steps in the dark reactions involve Calvin cycle C3 pathway, which is depicted in figure 12.10, and are mentioned as follows. Carboxylation. CO2 reduction starts with a 5-carbon sugar ribulose 1.5 biphosphate, that is denoted as RUBP. It is a 5-carbon sugar pentose with 2 phosphate groups attached to it. Glycolytic reversal. Molecules of 3-PGA form 1,3-diphosphoglyceric acid utilizing ATP molecules. These are reduced to glyceraldehydes 3-phosphate, that is 3-PGAL, by NADPH supplied by the light reaction of photosynthesis. For the Calvin cycle to run continuously, figure 12.9 denotes relation between light and dark reaction. It is also depicted by the chemical reaction as shown. Photorespiration. Photorespiration occurs under the conditions like high temperature, bright light, high oxygen and low CO2 concentration. It is a wasteful process linked with C3 cycle, where instead of fixation of CO2, it is given out. Figure 12.11 denotes photorespiration. C4 pathway or Hatch-Slack pathway. MD Hatch and CR Slack, while working on sugarcane, found 4 carbon compounds. Dicarboxylic acid as the first stable product of photosynthesis. It has been found to occur in tropical and subtropical grasses and some dicotyledons. Some of the important plants are sugarcane, maize, sorghum, etc. Figure 12.12 denotes the Karan's anatomy of C4 plant. CAM, Crassula cyan acid metabolism. It is one more alternative pathway of carbon fixation found in desert plants. It was first reported in the family Carassulaceae, so called as CAM. Figure 12.13 denotes C4 pathway. Factors affecting photosynthesis. External factor. First, light. It is an essential factor as it supplies the energy necessary for photosynthesis. Both quality and intensity of light affect photosynthesis. Highest rate of photosynthesis takes place in the red rays and then comes the blue rays. Carbon dioxide. The main source of carbon dioxide in land plants is the atmosphere, which contains only 0.3% of the gas. Under normal conditions of temperature and light, carbon dioxide acts as a limiting factor in photosynthesis. Graph 12.15 shows the effect of CO2 concentration. Third, temperature. Like all other physiological processes, photosynthesis also needs a suitable temperature. In the presence of plenty of light and carbon dioxide, photosynthesis increases with the rise of temperature till it becomes maximum. Fourth, water. Being one of the raw material, water is also necessary for photosynthetic process. An increase in water content of the leaf results in the corresponding increase in the rate of photosynthesis. Let's study the internal factors now. First one, though the presence of chlorophyll is essential for photosynthesis, but the rate of photosynthesis is proportional to the quantity of chlorophyll present. The final product in the photosynthesis reaction is sugar, and its accumulation in the cell slows down the process of photosynthesis. Blackman's law of limiting factors. The Blackman's law of limiting factors states that when a process is conditioned as to its rapidity by a number of separate factors, the rate of process is controlled by the pace of the slowest factor. Graph 12.16 denotes the light intensity and photosynthesis. The slowest factor is that factor which is present in the lowest or minimum concentration in relation to others. The law of limiting factor can be explained by taking two external factors such as carbon dioxide and light. Significance. This anabolic process uses inorganic substances and produces food for all life directly or indirectly. This process transforms solar energy into chemical energy. That is all in Chapter 3 Biology. Thank you.